Welcome back to the deep dive. Um, Today we are doing something a little different than our usual microbiology discussions.
Yeah, quite a bit different.
Usually we're looking at things you inhale, right?
Mm-hmm.
Or things you eat, the invisible threats just floating in the air that attack you systemically.
Right.
But today we are looking at things that have to be, well, they have to be pushed into you.
Pushed, stabbed, injected. We're leaving the surface. We're completely done with the
But
dermatophytes.
the ringworms.
Exactly. The ringworms and athlete's feet that just sort of sit on the keratin layer of your skin like moss on a rock. But we aren't quite at the level of the systemic killers either, the ones that target the lungs or the brain.
We're in the middle ground.
We are in the subcutaneous layer.
Like under the skin deep dive. And we are specifically tearing apart Chapter 63 of Murray's Medical Microbiology with, a few side glances at Jadwit's Chapter 45 to fill in some of the microscopic gaps. And the theme here, the thing that really connects every single organism we're going to talk about today, is violence.
It is. It's traumatic implantation. That is the phrase you are going to hear over and over again.
Which is really just a nice, sanitized medical way of saying you got stabbed by something.
Essentially, yes. Because these fungi, they're saprophytes.
They live in the dirt?
Right. They live in the soil, on plants, rotting wood. They have absolutely no interest in humans. They don't have the enzymatic machinery to dissolve intact skin like, say, some bacteria do.
They need a door.
They need a door. And that door is usually a thorn or a splinter, a bite, a cut.
I really like the analogy we discussed during our pre-read for this. If you think of the skin as a fortress wall, right? The dermatophytes are like the moss growing on the outside of the stones.
Right.
But these subcutaneous fungi, they are the invaders who just wait for a catapult stone to breach the wall. And once there's a crack, they rush in and build a fortified bunker just inside the gates.
That is a perfect visualization. And once they are in that bunker, the subcutaneous tissue, the fascia, the lymphatics, they really dig in. They are crawling.
They're incredibly slow, right?
Yeah, very slow. They don't kill you quickly. They occupy you. They resist the immune system not by hiding, but by building these massive structures. Granulomas.
Things the body just can't break down.
Exactly.
And for those of you listening who are prepping for boards, or maybe you just love a good medical buzzword, I've got to say, Chapter 63 is the undisputed heavyweight champion of buzzwords.
Oh, absolutely. It's the capital of buzzword country.
We're talking rose gardener, cigar-shaped yeast, copper pennies.
Cauliflower lesions?
Yes, Madura foot, Splendor hipply. If you're taking a pathology or micro exam, this section is just high-yield gold.
It really is.
So here's our roadmap for today. We are going to follow the Murray text structure exactly so you can follow along if you have the book in front of you. We got 5 main entities to cover.
The big 5.
First, the classic, Sporothrix or rose gardener's disease. Second, the visual nightmare that is chromoblastomycosis. Third, the limb destroyer, pneumococcal mycetoma.
That one is rough.
It really is.
Yeah.
Fourth, the creature feature, subcutaneous entomophthalmomycosis, also known as the hippo phase. And finally, the wastebasket category, subcutaneous phaeohyphomycosis.
It's quite a rogues gallery. But before we dive in, I want to emphasize one key concept for the listener. These are very distinct diseases.
Right.
In the systemic fungal section, everything kind of blurs together. It's all pneumonia, dissemination, fever. Here, the clinical presentations are radically different. A Madura foot looks absolutely nothing like a Sporothrix arm, so visualization is key today.
Okay, let's get our hands dirty. Or actually, maybe we shouldn't, considering the topic. Let's talk about Section 1, Sporothrix schenckii, the rose gardener's disease.
Absolute classic. This is the one every medical student learns first. The agent is Sporothrix schenckii. But Murray makes a point right in the first paragraph that we need to be careful with our taxonomy here.
Because we used to think it was just one species.
Right. But now we know it's a complex.
Molecular biology strikes again.
Exactly. It's the Sporothrix schenckii complex. They've split it up. You have S. schenckii sensu stricto, which just means in the strict sense. You have S. globosa, and then you have the one that is causing absolute havoc in South America right now, which is Sporothrix brasiliensis.
We are definitely going to come back to brasiliensis because that story is wild. But generally speaking, where does this fungus hang out?
It's ubiquitous. It's in the soil. It loves decaying vegetation. But the specific reservoir mentioned in every textbook, Murray included, is sphagnum moss.
Which is the stuff florists use to pack plants.
Yes. There was actually a massive outbreak in the 1980s involving forestry workers. They were handling all these pine seedlings that were packed in sphagnum moss, and hundreds of people got infected.
Wow.
So while we call it rose gardener's disease because of the rose thorns, it could just as easily be pine seedling packer's disease.
It just doesn't roll off the tongue quite as well.
No, it doesn't, but the mechanism is exactly the same. You have the fungus on the plant, you have a sharp object, the thorn, the pine needle, whatever. It pricks the skin, and the fungus is injected directly into the dermis.
Okay, so I'm the gardener. I've been pricked. The fungus is in. Now, Sporothrix is a thermally dimorphic fungus. We've seen this term before in other deep dives, but let's explain what it means in this specific context.
Sure. Thermal dimorphism is really the hallmark of the major fungal pathogens. It just means the fungus has two totally different shapes depending on the temperature. The rule you absolutely have to memorize is mold in the cold, yeast in the heat.
Mold in the cold, yeast in the heat.
Exactly. At 25 degrees Celsius, so room temperature, or out in the garden, it grows as a mold.
Yeah.
And if you look at it under a microscope from a culture, the hyphae are narrow and septate. And the conidia, the spores, are arranged in this really beautiful pattern. Marie calls it a rosette or a daisy petal formation.
It's iconic. You look at the Jowett's plates, you see this little thin stalk with a flower-like cluster of spores right at the tip.
Right, but that is not what makes you sick. When that spore gets pushed into your warm body, 37 degrees Celsius, it undergoes a phase transition.
It realizes it's not in the soil anymore.
Exactly. It stops being a mold, it transforms into a yeast.
And this is the famous cigar shape.
Yes, this is the high-yield visual for the exam. They are pleomorphic, meaning they can vary a bit in shape, but the classic textbook description is an elongated, cigar-shaped yeast. It buds, but often with a very narrow base.
So walk us through the infection timeline. I got pricked by the rose thorn 2 weeks ago. What am I seeing on my hand right now?
It starts as a small papule at the exact site of the injury. It's usually painless at first. You might just think it's a bug bite or a splinter, but then it starts to grow. It becomes a nodule, and eventually it ulcerates, it breaks open, and starts draining a little bit of fluid.
Now, if it just stayed there, it'd just be a weird chronic sore.
Yeah.
But Sporothrix travels.
It does. And the way it travels is unique. It doesn't usually go into the blood. Hematogenous spread is pretty rare here. It uses the lymphatic system.
The body's drainage pipes.
Exactly. It enters the lymphatic vessels and starts moving proximally, so up the arm toward the armpit. And as it hits the lymph nodes along the way, those nodes swell up, get infected, and become secondary nodules.
So clinically, you're looking at a line of red, bumpy nodules tracing a visible path up the patient's arm.
It looks exactly like train stops on a map. This is called ascending lymphangitis. And honestly, if you see a patient with a sore on their hand and a line of red bumps going up their arm, Sporothrix should be the very first word out of your mouth.
But is it the only word? Because Murray lists a differential diagnosis here that we should mention.
It's not the only one, no. There's a classic mimicker, Mycobacterium marinum.
The fish tank granuloma.
Right. M. marinum is a bacteria, cousin of tuberculosis. It lives in water. So if you are cleaning a fish tank and you have a cut on your hand, you can get it. And it can cause that exact same sporotrichoid spread.
The ulcer on the hand, the line of Barron, bumps up the arm.
Yep. So the patient's history is everything here. Did you prick your finger on a rose, or were you scrubbing algae out of an aquarium?
But even then, history can trick you. Murray actually details this fascinating case study of a fisherman in Brazil.
Oh, right, that case.
He gets stuck by a fish spine, develops the ascending nodules. The doctors naturally assume it's Mycobacterium marinum because, well, he's a fisherman. He got stuck by a fish.
It makes perfect sense.
Right, but the antibiotics don't work.
Because it was Sporothrix.
Exactly, it was Sporothrix. The fish spine carried the fungus, or maybe the organic matter in the water did. So the lesson is, you can't rely 100% on the rose versus fish history. You really need a biopsy.
You do. You have to look at the tissue.
Let's talk about that biopsy. What are we actually looking for? We mentioned the cigar yeast. Are they easy to find?
Surprisingly, no. In human tissue, Sporothrix yeast are notoriously scarce. You can stain the tissue and look for hours and not see a single cigar.
That seems incredibly frustrating for a pathologist. So what do you look for instead?
You look for the body's reaction to the fungus. And there is a specific structure, another major buzzword, called the asteroid body.
The asteroid body. Sounds cosmic.
It looks cosmic. If you see it on a slide, imagine a central yeast cell. And sometimes you can't even see the yeast itself clearly, but it's surrounded by these radiating rays of pink eosinophilic material. It looks like a starburst.
And what is that stardust exactly?
It's the Splendor-Heppley phenomenon.
Splendor-Heppley.
Yes. That pink material is actually antigen-antibody complexes. It's the human immune system trying to entrap the fungus, coating it in antibodies and cellular debris. It's beautiful under the microscope, but functionally it's a sign of a stalemate.
The fungus is trapped, but it's not dead.
Exactly.
Now, before we get to the treatment, we have to circle back to Sporothrix brasiliensis. You mentioned earlier it's causing havoc. Why?
This is such a fascinating evolutionary jump. Standard Sporothrix is environment to human. You touch a plant, you get sick. But S. brasiliensis has adapted to be a true zoonosis. In Brazil, it is spreading like wildfire among stray cats.
So the cats are the vector.
The cats are the reservoir and the vector. They get these horrible ulcerated lesions on their faces and their paws. They fight each other, spreading it cat to cat. And then a human tries to help the sick cat, or maybe just walks by and startles it and gets scratched or bitten.
And Murray points out that this species is significantly more virulent.
Much more. The fungal load in the cat wounds is incredibly high, so when you get scratched, you are getting a massive inoculum injected right into your tissue. The disease tends to be more severe, and there are way more cases of it disseminating to other parts of the body, beyond just the lymphatics. It's become a major public health crisis in places like Rio de Janeiro.
So note to self, if you are in Brazil, beware the stray cats.
Absolutely.
Let's talk about treating this. How do we kill the rose gardener's fungus?
Well, we have the old school way and the new school way. The old school treatment is potassium iodide, specifically a saturated solution of potassium iodide, or schenckii.
This has always baffled me. Iodine, like the brown stuff we put on cuts, but we drink it.
You drink it, you put drops of it in juice or milk, and the crazy thing is, it works. We actually don't even know the exact mechanism. It seems to either enhance the immune response or maybe act directly on the fungal cell wall, but the side effects are rough.
I can imagine.
It tastes like metal, it causes severe gastric upset, and weirdly, it can cause your salivary glands to swell up massively.
The chipmunk sign.
Yes, you look like a chipmunk.
I assume we have something better now.
I do, thankfully. Itraconazole. It's an oral antifungal. It is highly effective for lymphocutaneous sporothrix. That is the gold standard of care now.
There's a really interesting note in Murray about heat.
Yes. Think back to the dimorphism. It prefers the cold mold form, right? It struggles to maintain that yeast form at high temperatures. So applying local heat pockets, literally just hot packs, to the infected arm can actually help inhibit fungal growth and speed up the healing.
That is such a cool low-tech biohack. You just use the organism's own biology against it.
Exactly. It's elegantly simple.
All right, let's move to section 2. We are leaving the rose garden and heading deep into the tropics. Chromoblastomycosis. Let's break down that name first. Chromo means color. Blasto usually refers to budding or yeast. Mycosis means fungal infection.
Spot on. This is a chronic fungal infection characterized by two main things: pigmented or colored fungi and warty, cauliflower-like lesions on the skin.
The visual description in Murray is intense. They call it cauliflower-like.
It is genuinely disturbing to look at. These lesions start as small, scaly papules, almost always on the legs or feet of barefoot agricultural workers. But over years, and I mean years, this is an incredibly slow process, they grow, they pile up.
They hyperkeratinize.
Yes, they become verrucous, which means warty. They literally look like distinct florets of a cauliflower growing out of the person's leg.
And who are the culprits here? Is it just one fungus?
No, and this is a key concept for this entire chapter. Chromoblastomycosis refers to a clinical syndrome. It's the presentation. It can be caused by several different fungi. The most common ones you see in the text are Fonsecia, Cladophilofera, and Filophorid.
Those are absolute mouthfuls. Do we need to memorize those specific genus names for the boards?
For a specialist micro exam, yes. But for the generalist, you really need to know the group name. Demachiaceous fungi.
Demachiaceous, let's define that.
It comes from the Greek word for bundle or bandage, but in mycology, it practically means dark. These fungi naturally produce melanin in their cell walls. If you grow them in a petri dish, the colonies aren't white or fuzzy. They are black, olive green, or dark brown. They look like dark velvet.
And that melanin isn't just for show. It's not cosmetic.
No, it is a primary virulence factor. Melanin is incredibly tough. It absorbs free radicals. So when our white blood cells try to destroy the fungus with an oxidative burst, the melanin just absorbs it. It acts like armor plating.
Which explains exactly why these infections are so chronic and so hard to clear.
Exactly. The body tries to kill it, fails, and just builds more scar tissue around it instead.
Now, Let's talk about the #1 exam question for chromoblastomycosis, the microscopic diagnostic feature.
The copper pennies.
The copper pennies, also known as chlorotic bodies, medullary bodies, or muriform cells.
You have to know all those names. They will test you on any of them.
Describe what we are actually looking at under the scope.
So you take a skin scraping or a biopsy from one of those black dots on the cauliflower lesion. Under the microscope, you see these round, thick-walled cells. They are chestnut brown, and you don't even need to stain them to see that color because of the melanin. And inside the cell, you see septations, lines running both horizontally and vertically.
Like a window pane.
Yes, or a hot cross bun. That internal division in two planes is completely unique. If you see those brown window pane cells, stop. You have the diagnosis. It's chromoblastomycosis. It doesn't matter if the underlying fungus is Fonseca or Phyllophora. The body itself is the diagnosis.
Why do they form these specific bodies?
It's their tissue form. They don't form hyphae in the tissue, and they don't form typical budding yeast either. They form these sclerotic bodies to withstand the intense host pressure. They are in pure survival mode.
And what happens to the patient if this goes untreated?
It gets ugly. The lesions can spread to cover the entire lower leg. Secondary bacterial infections are super common. And because there's so much inflammation and continuous scarring, the lymphatic vessels in the leg get crushed. You get what's called secondary elephantiasis.
The leg just swells massively.
Yes, the leg swells up to huge proportions. It's highly disfiguring and disabling.
How do we treat it? If they have that melanin armor, I imagine it's resistant to drugs.
It's very tough. Treatment is prolonged. We're talking months to years. We use itraconazole or terbinafine. Often we have to add flucytosine to try to boost the killing power, but relapse is very, very common.
Can you just cut it out if it's on the surface, just excise it?
Only if it's very small and caught early. But Murray gives a very specific warning about surgery here. If you don't get wide enough margins, the fungus can recur right inside the surgical scar.
Oh, wow.
And worse, the trauma of the surgery can actually trigger the infection to spread via the lymphatics if you disturb the tissue too much.
So catch it early, treat it along with antifungals. Let's move to section 3, the Madura foot, or more formally, eumycotic mycetoma. Now, before we get into the pathology, I need you to clarify a major point of confusion for a lot of students here. Mycetoma sounds like a specific disease, but there are actually two types, and they are caused by totally different kingdoms of life.
This is the most critical distinction in this entire section. If you take nothing else away from this deep dive, take this. Mycetoma is a clinical syndrome. It describes a swollen, deformed foot with draining sinuses. But that syndrome can be caused by fungi, which is eumycotic mycetoma, or by a bacteria, which is actinomycotic mycetoma.
Eu meaning true. So eumycotic is true fungus, and actinomycotic is caused by actinomycetes, which are filamentous bacteria. Why does this distinction matter so much clinically?
Because the treatment is polar opposite.
Right.
If it's bacteria actinomycetoma, you give them antibiotics. Bactrim, streptomycin, and the patient usually gets cured. They keep their foot. But if it's fungal eumycotic antibiotics, do absolutely nothing. And antifungals are barely effective at all. The definitive treatment for eumycotic mycetoma is often amputation.
Wow. So misdiagnosing this could literally cost someone their leg.
Literally. You have to know which one it is before you treat.
So how do we tell them apart? Let's start with the clinical picture. Murray talks about a clinical triad for mycetoma.
Yes. Regardless of whether it's bacterial or fungal, it presents with these three classic signs. 1, tumefaction. That's just a fancy clinical word for swelling. The foot, or sometimes the hand, is swollen, grossly distorted, and feels woody hard. 2, draining sinuses. You see these little tracts opening to the skin surface, oozing pus and fluid. And 3, granules. This is the money sign. Granules, or grains, actually come out of those sinuses.
Let's double-click on the grains. These aren't like grains of sand, right?
No. A grain is actually a macroscopic microcolony of the organism itself. It's a tight, dense ball of fungal hyphae or bacterial filaments if it's actinomycotic cemented together by a hard matrix.
It's like the organism built its own concrete bunker to live inside the tissue.
Exactly. And the color of that grain gives you a massive diagnostic clue.
Okay, break down the colors for us.
For eumycotic or fungal mycetoma, Madarella mycetomatis is the most common cause worldwide. It produces black grains. Cedosporium or Fusarium, which are also fungi, produce white or pale grains.
So if I'm examining a patient, I squeeze the sinus tract and a black speck comes out.
It's almost certainly fungal, Madarella. Bacteria very rarely produce black grains.
But if a white speck comes out?
Then it's tricky. It could be fungal, like Cytosporium, or it could be bacterial, like Nocardia. So if it's white, you have to crush that grain, put it on a slide, and look under a microscope.
And what are we measuring under the scope?
The size of the filaments. If you see broad, thick hyphae greater than 4 or 5 microns wide, it's a fungus. If you see tiny, thin, thread-like filaments that are less than 1 micron wide, it's bacteria.
It seems simple enough in theory, but in a rural clinic in the field, I imagine it's messy.
It is very messy, but that simple crush prep determines if the patient gets a course of pills or an amputation.
Let's talk about the destruction. Why is Madura foot so feared?
Because it respects absolutely no tissue boundaries. Most skin infections stay in the skin or the fat. Mycetoma burrows. It goes through the dermis, it eats through the fascia, into the muscle, and eventually it invades the bone itself.
This just dissolves it.
It creates these olytic lesions in the bone. It turns the architecture of the foot into a non-functional Swiss cheese-like mass of scar tissue and fungal bunkers.
And the U variety, the fungal one, is just totally resistant to drugs.
The fungus inside those grains is metabolically very slow, and it's protected by that cement matrix. Antifungals just can't penetrate deep enough or fast enough. We try itraconazole or voriconazole to shrink it, maybe slow it down, but to truly cure it usually requires cutting out all the infected tissue. And since it's often deep inside the foot bones, that means amputation below the knee.
That is a heavy diagnosis.
It's completely devastating for the patient, especially since it usually affects young men who are the primary laborers for their families.
Okay, take a breath. That was heavy. Let's move to section 4. Now, the name here is an absolute tongue twister. Subcutaneous Mycoses.
Try saying that 3 times fast.
No, thank you. Taxonomically, we're shifting gears here. These aren't the demaciaceous fungi we just talked about. These are mucormycetes.
Right. Now, when doctors or students hear mucor, they usually panic.
Because they think of rhizopus.
Exactly. They think of the black mold that eats the faces of diabetics in ketoacidosis. That is systemic mucormycosis. It invades blood vessels, it's angioinvasive, and it kills you in a matter of days.
But this entomophthalmomycosis is different.
Very different. They belong to the order Entomophthalus. They are not angioinvasive. They don't eat blood vessels. They don't race to the brain. They are slow, chronic growers that just sit in the subcutaneous fat.
And we have 2 main characters here. A tale of 2 patients, really. Let's introduce patient A.
Patient A is an adult. The organism is Conidiobolus coronatus, and the infection is rhinofacial.
Which means nose and face.
Yes. It likely starts by inhaling the spores, or maybe picking the nose with contaminated fingers. The fungus infects the nasal sinuses and the soft tissue of the upper lip.
And the visual here is the hippo face.
It's tragic. The intense inflammation causes this massive, firm, painless swelling of the upper lip and nose. It puffs the face out entirely. It creates a deformity that looks like a hippopotamus snout. And it's hard and woody to the touch because of all the fibrosis.
But it doesn't rot the face off like the other mucor? It doesn't necrose?
No. It just distorts it. It pushes things around. It can close off the airway just by sheer bulk, but it doesn't destroy the tissue necrosis style.
Now, patient B.
Patient B is a child, usually a young boy. The organism here is Basidiobulus ranarum. This is a distinct presentation. It doesn't affect the face at all. It affects the proximal limbs, so the thighs, the buttocks, the back.
And the texture is described differently. Rubbery.
Yes. It forms a disc-shaped mass under the skin. It feels like a hard frisbee or a rubber puck that you can actually move around slightly. It's not fixed to the bone. It's just sitting there in the fat.
Is there a specific risk factor for the kid? Murray mentions dung.
Yeah, this is interesting. Basidiobulus is commonly found in the GI tract of reptiles and amphibians. So lizards, frogs, the spores come out in their feces. Kids playing in the dirt, maybe interacting with lizard dung. That's the suspected vector for traumatic implantation here.
Note to parents, wash your kids' hands after they play with lizards.
Good advice.
Microscopically, what connects the hippo face and the rubber thigh? They look so different clinically.
The Splendor-Hupley phenomenon again, just like we saw in Sporothrix. If you biopsy these masses, you see broad, ribbon-like hyphae, which is typical of mucor, but they are surrounded by an intense sleeve of eosinophilic material.
Eosinophils usually mean parasites or allergies. Why are they here for a fungus?
That's a characteristic of this specific fungal order. They trigger a massive eosinophilic response from the host. The granulomas are just packed with eosinophils.
What about treatment? Please tell me it's better than the Madura foot.
It is much better. These respond surprisingly well to itraconazole, and interestingly, potassium iodide works here too, just like in Sporothrix. The masses usually shrink down. However, for the hippoface caused by condylobolus, the fibrosis, all that dense scar tissue might stay.
So the fungus is dead, but the face is still distorted.
Exactly. Even after the infection clears, the deformity remains, and they might require extensive plastic surgery to reconstruct the airway and the lip.
We have reached the final section, Section 5. Subcutaneous pheohyphomycosis. You called this the wastebasket in our prep notes. Why?
Because it's essentially a diagnosis of exclusion. Let's break down the word first. Pheo means dark, like dusky. Hypho means hyphae. Mycosis means fungus, so it's an infection by dark fungi.
But wait, we already did dark fungi. Chromoblastomycosis is dark, and mycetoma had black grains.
Exactly, and that's the diagnostic rule you need to follow. If you have a dark fungus A and D, you see sclerotic bodies, the copper pennies, then it is chromoblastomycosis. If you have a dark fungus A and D, you see grains, then it is eumycotic mycetoma. But if you have a dark fungus and you do not see pennies, and you do not see grains, it goes in the wastebasket. It is pheohyphomycosis.
Ah, I see. It's defined by what it lacks.
Precisely. In the tissue, you just see dark, irregular hyphae, maybe some yeast-like cells mixed in, but no organized structures, no bunkers, no pennies.
Who are the agents?
It's a huge list of environmental molds. Exuviala, Alternaria, Curvularia, Pheoacromonium. These are molds that are absolutely everywhere. They are on your shower curtain. They are on rotting wood, in the soil.
And what is the clinical presentation? How does the patient walk in?
The classic presentation is the pheohyphomycotic cyst. Imagine you get a splinter in your finger or your foot. The splinter has the fungus on it. The skin heals over it, but the fungus starts growing slowly inside.
The body walls it off.
Yes. You end up with a single, firm, encapsulated cyst right under the skin.
It probably feels like a ganglion cyst or a boil.
Exactly, and that's almost always what the doctor thinks it is. They go in to excise a supposed ganglion cyst, they cut it out, send it to pathology, and the pathologist calls back and says, hey, the center of this cyst is necrotic and there are brown fungal hyphae lining the entire wall.
And often they find the smoking gun right in the middle.
Yes. Frequently, they find the actual wood splinter still inside the cyst. It's microscopic proof of the traumatic implantation.
Murray has a specific case study here about a transplant patient.
Yes, a diabetic renal transplant patient. He developed a lesion caused by pheoacromonium, but he didn't recall any trauma at all.
What does that tell us?
It highlights a really important point. In immunocompetent people like you and me, these fungi usually just make a walled-off cyst and stop. But in immunocompromised people transplant recipients, HIV patients, these wastebasket fungi can be much more aggressive.
They don't stay in the cyst.
Right. They can spread locally, they can turn into massive abscesses, they exploit the weakened immune system.
But for the standard cyst in a healthy person, what's the treatment?
The surgery is the cure. If you excise the cyst intact, if you don't rupture it and spill it in the wound, you have removed the entire infection. You often don't even need to prescribe antifungal pills.
That is easily the happiest ending we've had today.
It is. Compared to amputation or severe facial deformity, a simple outpatient excision is a massive win.
So we've covered the big five. Let's zoom out for a second and synthesize this. We have Sporothrix, the traveler. It uses the lymphatics.
Right.
We have chromoblastomycosis, the builder. It builds cauliflower towers and copper penny bodies. We have mycetoma, the burrower. It drills deep into the bone with grains. We have endomorphomycosis, the inflamer. It puffs up the face or thigh with the eosinophils. And pheohyphomycosis, the cyst maker.
That is a phenomenal summary. And remember the unifying theme. The skin barrier is crucial. These things only get in when we let them in via trauma, and once they are in, they are stubborn. They are adapted to survive in harsh environments in the wild, so the human immune system is another harsh environment to them. They bunker down.
Before we get to the quiz, I have to ask about the overarching lesson here. We talk a lot about traumatic implantation. Does this change how you live your life? Do you wear gardening gloves religiously now?
I absolutely do. Gloves and closed-toe shoes. If you look at the global burden of mycetoma and chromoblastomycosis, it is overwhelmingly concentrated in populations that work barefoot in agriculture. Simple protective gear shoes and gloves would prevent 99% of these infections. It's fundamentally a disease of exposure.
I also want to leave the listener with a provocative thought based on what we've covered. We usually think of these environmental fungi as accidental tourists in the human body. They don't mean to be here.
Right, they prefer the soil.
But look at Sporothrix brasiliensis. It has evolved to utilize mammalian behavior catfights, biting, scratching to spread. It's moving from accidental implantation to active zoonotic transmission. It makes you wonder if these accidental tourists are slowly adapting to use us as their primary ecosystem.
It's a chilling thought, but the evolutionary pressure is definitely there. We are warm, nutrient-rich environments walking around the Earth.
All right, are you ready to test our listeners? Let's see if those buzzwords stuck.
Let's do it. I've prepared 6 questions. These are single best answer style.
I'll read this scenario. I'll pause for a few seconds. So, shout the answer at your windshield or your dog, and then our expert will give us the answer in the why.
Sounds good.
Question 1. A 35-year-old man presents with a painless ulcerated nodule on his right index finger and several similar nodules tracking up his right forearm. He mentions he is an avid gardener. A tissue biopsy is performed. Which of the following microscopic findings would be most characteristic of the causative agent in the tissue? A. Broad, non-septate hyphae. B. Copper-colored septate spherical bodies. C. Cigar-shaped yeasts. Or D. Spherules containing endospores.
The answer is C, cigar-shaped yeasts.
Tell us why.
The clinical scenario is classic sporothrix. You've got the rose gardener history and the ascending lymphangitis. The question specifically asks for the finding in the tissue, which means 37 degrees Celsius. At that temperature, it's the yeast form, which is cigar-shaped. A is mucor, B is chromoblastomycosis, and D is coccidioidomycosis, which is systemic.
Excellent. Question 2. You're examining a skin scraping from a verrucous cauliflower-like lesion on the foot of a tropical farmer. You see brown, round, thick-walled cells with internal horizontal and vertical septations. What is the clinical name of this disease?
The answer is chromoblastomycosis.
The breakdown.
The description is exactly that of sclerotic bodies or medlar bodies. The brown dividing window panes, these are pathognomonic for chromoblastomycosis, and the cauliflower appearance on the foot supports it perfectly.
Moving on to question 3. A patient presents with a swollen, deformed foot with draining sinuses. Examination of the exudate reveals black granules. A diagnosis of eumycotic mycetoma is made. Which genus is the most likely cause? A. Actinomydera, B. Nocardia, C. Madarella, or D. Sporothrix?
The answer is C, Madarella.
Why Madarella?
We know it's eumycotic, meaning fungal, because of the black grains. Madarella myxotomitis is the classic, most common cause of black grain fungal mycetoma. Actinomydera and Nocardia are bacteria. They cause actinomycetoma, and Sporothrix doesn't cause mycetoma or grains at all.
Okay, question 4. A 12-year-old boy presents with a firm, rubbery, palpable mass in the subcutaneous tissue of his left thigh. He has no fever. Biopsy shows a granuloma rich in eosinophils and broad postiseptate hyphae surrounded by splendor-hepli material. What is the likely agent? A. Conidiobolus coronatus, B. Basidiobulus ranarum.
C. Sporothrix schenckii, or D. Rhizopus oryza.
The answer is B. Basidiobulus ranarum.
And how do we differentiate that from the others?
Well, the patient is a child, and the location is the proximal limb, the thigh. Conidiobolus, option A, typically affects the face and nose in adults. The histology with the eosinophils and splendor-hepli fits entomophotomycosis perfectly. Question 5. A renal transplant patient develops a solitary cyst on his shin. The cyst is excised. Pathology reveals dark, irregular fungal hyphae within the cyst wall, but NO sclerotic bodies and NO grains are seen. What is the most appropriate classification for this infection?
The answer is subcutaneous pheohyphomycosis.
End of the wastebasket.
Exactly. It is a dark or dimaceaceous fungus, but it lacks the specific diagnostic structures of chromonosclerotic bodies, and it lacks the grains of mycetoma. Therefore, by exclusion, it falls into the pheohyphomycosis category.
Last one. Question 6. Let's throw a curveball. Regarding Sporothrix, which form is observed when the fungus is grown on Sabrod agar at 25 degrees Celsius? A. Cigar-shaped yeast. B. Sclerotic bodies. C. Mold with conidia in a rosette pattern.
Because of the temperature.
Precisely. Sporothrix is thermally dimorphic. At 25 degrees Celsius, which is room temperature or culture conditions, it grows as a mold. The rosette or daisy-pale conidia arrangement is the classic textbook description. The cigar yeast is only found at body temperature. And there you have it. You have survived the subcutaneous layer.
We finally made it through the fortress wall.
I highly, highly recommend opening Murray chapter 63 and just looking at the pictures while this is fresh. The visual of the copper penny and that Madura foot will stick in your brain way better than just hearing our voices describe them.
Absolutely. Visuals are everything in dermatology and mycology.
Thanks for diving deep with us today. Stay safe, wear thick shoes in the garden, and we'll see you in the next layer.
Stay curious.